Okay, so what I'm going to focus on today is we're going to talk about DNA repair mechanisms, and I'm specifically going to talk about DNA break repair. So in my research lab, we use bacteria as a model organism for understanding basic cellular processes. And so what you can see here, or the goal of this slide is, so you have a single cell here, and it has a cell wall, which is made out of a particular type of carbohydrate, and then on the inside of that, it has a plasma membrane, and that membrane is made up of lipids, and it allows for the selective passage of materials in and out of the cell. And then inside of the cell, you have the cytoplasm, which has all kinds of biological molecules. And then there's also the DNA, which is this thing that looks like a ball of yarn. Okay. And so in this case, we have a cell, and it's duplicating its DNA. After that DNA is duplicated, then the chromosomes are segregated to opposite sides of the cell, and then the cell goes through the process of cell division. And so what you can see here is you can see the cell membrane beginning to pinch in, and then as this process continues, the cell membrane will be complete, and then two cells will be generated. And so this is simply the propagation of life on a basic level in thinking about bacteria. And then the bacteria will go on and do this again, provided that they have sufficient nutrients to grow, replicate their DNA, and go through that process. And my lab is very interested in understanding how the DNA will ultimately regulate this process. So in thinking about DNA, I just want to start at the beginning and start with the basics so that we're all on the same page as we move forward, because it's difficult to think about how it would be repaired if we don't have a sense for what the molecule is. And so DNA is double-stranded, usually. Okay, that's its most stable form. And what you have are phosphate groups and sugars that make up the phosphodiester backbone. And then pointing inside of the double helix are bases. And the bases are nitrogenous bases. They have quite a bit of nitrogen. And then they base pair together. And so you have, for example, cytosine and guanine, and these are forming hydrogen bonds between the bases. And then thymine and adenine, and they are adenosine, and they are forming hydrogen bonds between those bases. And so what happens is the bonds that form in the backbone of the DNA are very stable, and that's a covalent bond. It's not easy to break. And then the bonds that form between the bases are not as strong, and those are called hydrogen bonds. But when you have thousands and thousands of those bonds, then they can become stable as the DNA is annealed. And so one of the major properties here is that C always pairs with G, and T always pairs with A. And so if I were to take away one of these strands, you would know what the sequence of the other strand is, because you would know that the G has to pair with a C and the A has to pair with a T. Okay? Because those are the complementary base pairs, and we're going to discuss this a little bit, and those are the only pairs that will work properly. Okay? And so if we look at the model here, so now we have a nice model representation of the double helix, and you can see on the outside we have the phosphodiester backbone, which is the phosphate and sugar backbone, and then pointing on the inside we have the bases that are then hydrogen bonded together, and it takes on this helical representation. Okay. So within the cell, we have to, as we're thinking about DNA, and we're going to talk about DNA repair, we need to think a little bit about the consequences if DNA were to not be repaired properly. And so we just want to think about the process as it normally functions. And so DNA is storing the genetic information for all cells. Okay? And so this is a very stable storage medium. And then DNA is converted to another nucleic acid called RNA during the process of transcription. And so the message on the DNA will be transcribed and converted into RNA. All right? And then RNA, which is a nucleic acid like DNA but different, will then be translated into protein. And so it's a conversion of the message from nucleic acids to amino acids, and amino acids make up protein. And then those proteins will go off and do whatever their function happens to be. And so if we just look at the flow here, we have our DNA, and then using the base pairing rules that we just discussed, RNA will be, an enzyme called RNA polymerase will copy the DNA information into this RNA, which is a messenger RNA, and this is an unstable molecule. And then there's a complex called a ribosome, which is another type of RNA, as well as proteins that will decode this information and convert it into a protein. And then the protein can go off in the cell and do whatever function it has to allow for the cell to do whatever the cell needs to do. Okay? And so it's this flow of information, right? We have information stored in DNA. The information is then transcribed into a messenger, and then that messenger is translated into a protein which will carry out its function. So that is the flow of information within a biological system. Okay. So obviously the DNA is pretty important because it contains that blueprint which is important for life, right? It has the genetic information that's necessary for the cell to do what it needs to do. And so I want to focus a little bit more about the synthesis of DNA, and then we'll ultimately get into the repair of it. And so in bacteria, many bacteria have a circular chromosome, which is shown here. You have linear chromosomes. Okay? And for a bacterial chromosome, there's a single origin of replication, meaning there's a place on the chromosome where DNA synthesis will begin. And then there is a site about 180 degrees from the origin where DNA synthesis will be terminated or will stop. And so what I'm showing you here is the process of DNA replication on a larger scale, where we're looking at it for a whole chromosome. And so, you have basically once the DNA double helix is unwound, then each strand will serve as synthesis for the new strand, right? Because based upon the base pairing rules, once you've opened up that double helix, then you can copy either strand because of the base pairing rules that exist. G pairs with C and A pairs with T. If we look at it from a chemical process and just the way that it occurs, here we have, because DNA strands are anti-parallel, meaning that you have a 5' end of a phosphate and then a 3' end of the sugar, and so one strand is going 5' to 3', the other strand, which is paired with, is going 5' to 3', so they're anti-parallel. Okay? And so what happens is with the DNA strands are made up of single nucleotides, and the DNA polymerase is responsible for making these DNA polymers. Okay, so the DNA polymerase will bind to an incoming nucleotide, and then the nucleotide has 3 phosphates, and it's the breaking of these bonds which allows for the formation of a new phosphodiester bond and the release of phosphates, and that release of energy is what ultimately drives this reaction. And it's very quick. And so, for example, the DNA polymerase in bacteria can replicate about 750 nucleotides per second. Okay? So it's a very fast, it's a very robust enzyme. And it does it very, very accurately. Okay? And so it makes, in the organism that we study, it makes a mistake once every 59 million base pairs. So the system is very good at what it does. And that's something that's fascinated me throughout my whole career, and that's one of the reasons that I study this process. All right. So those are the basics for DNA synthesis. What you want to take away from this, in large part, is the complementary base pairing. Okay? Because that's going to be very critical to ultimately repair something if it's broken. All right. Another feature is that cells have lots of DNA. And so this is a bacterial cell called E. coli, and somebody has opened it up a little bit and allowed for all the DNA to spill out, right? And so this that circular chromosome that I showed you in a cartoon format, this is that circular chromosome when it's been allowed to leak out of the cell. Okay, so you can see that that's a lot of DNA that needs to be packaged within the cell. Okay. And then we can take another view of this when the DNA is actually in the cell. And so this is a microscopy image that I took many years ago. And so what I've done is I've used a dye that will stain the membrane, and then a blue dye that will stain the DNA. And you can see that the DNA, for the most part, fills the entire cell. It takes up a large volume within that cell. All right. Now, in eukaryotic cells like our cells, and there's more There's much more compartmentalization. You have membrane-bound organelles. The DNA is contained within an area called the nucleus. However, in a bacterial cell, the DNA takes up a very large fraction of the volume within that cell. Okay. And so, again, during my career, I've been very interested in how that DNA is duplicated with such high accuracy, as well as when it's broken or if there's something wrong with it, how does it get repaired? Okay. So let's talk a little bit about DNA breakage. What happens when things go wrong? Right. So for all of you, when you're sort of sitting somewhere breathing or sleeping and breathing, you are generating reactive oxygen species, okay, which is your cells going through normal metabolism will generate reactive oxygen species. And those will cause what we consider spontaneous types of damage. And those can be often in the form of base damage sites where we'll attack one of the bases, one of the nitrogenous bases within the double helix and damage that site. Okay, and that needs to be repaired. So for example, let me back up here. When the DNA polymerase is synthesizing, if it gets to, let's say we have adenosine here, if that were to be modified chemically in some way, then the DNA polymerase would have a more difficult time pairing something across from it because it wouldn't be able to fit it correctly. Okay? And that's where the damage becomes a major problem, is when the DNA is damaged and that template serves for synthesis of the new strand. If the base is correct, is normal, then it can replicate that with very high accuracy. If that base is damaged, then the DNA polymerase has a very difficult time trying to pair something with it because the hydrogen bonding that occurs will not be correct, okay, as one example. Okay, so that's a type of DNA damage. And then obviously there are things that we encounter in the environment, such, X-rays, this is one example in a doctor's office, where that can result in DNA breaks because it has enough energy to either, which I'll talk about, either ionize water or to damage directly through direct contact with the DNA. Okay, so there's a variety of different types of damage that can occur, and so what I'm showing you here is a single-stranded break, which is a break within the phosphodiester backbone, that backbone of the DNA. We can have bases that get cross-linked together, so there can be a chemical modification that covalently links bases together. Okay. We can have a missing base where one of these bases is gone. All right. And so if that base is gone and then that serves as a template for synthesis, well, there's nothing to pair with, right? What is the DNA polymerase going to do? And then there can be very large modifications that hang off of the bases. And then we can have, and what we're going to talk about a lot today, or for the most of this lecture, will be a double-stranded break where both of the phosphodiester backbones are broken and the DNA is now in two pieces. Okay. So how often does damage occur? So these are averages, but this is something to think about a little bit, in that there are about 30,000 sites in the DNA that are damaged in each one of your cells during a 24-hour period. Okay. And so during the course of this lecture, each one of your cells will have to repair about 1,250 sites of damage. Okay, and it has nothing to do with what I'm saying, right? Or, it's independent of me. It's just you living and breathing and sitting here. And then this has nothing to, and we're not thinking about if you go out outside without sunblock, right? I always sunblock my children before they go out to the beach, and they're not happy with that. But the amount of damage that cells incur by UV light is tremendous, okay, and far exceeds these numbers. But this is just due to the normal byproducts of cellular metabolism, which are reactive oxygen species. Okay. So as far as DNA break repair goes, and just to be clear, with the 30,000 sites, those are not breaks. Those are most of those lesions. Breaks make up a very small percentage of the damage that your cells would have. And one of the reasons, and it's a good reason, is it's very difficult for the cell to fix that, frankly. And a lot of the other types of damage are much easier to fix. And it requires quite a bit to break both strands of the DNA. Okay. But cells have three basic types of double-strand break repair. One is called non-homologous end joining. The other is called single-strand annealing. And then the third is called homologous recombination. And so I just want to go through those types of repair so you have a sense for the different ways in which your cells and many cells can repair broken DNA. All right. So here we have these two lines here that I'm pointing out represent the two strands of DNA, and something happens and the two strands are broken. And so in a very simplified form, what happens is there are repair proteins that will recognize the ends of the break. Okay? And then they will bring in other proteins that will then stick it back together. All right? And so what happens is, depending on how this DNA is broken, these proteins may need to change the ends of it to make them compatible to put it back together. All right? And so one of the problems, this is a great repair pathway, and it's very important. One of the problems, however, is that, you can think about this for a minute. If the ends are not completely complementary to go back together easily, and the proteins need to modify them to put them back together, then what happens? You're going to have potentially a loss of information in that coding, if it occurs in a coding sequence. Wherever it occurs in the DNA, you have the potential for a loss or a change in that base pair information. Okay? So it solves the initial problem of taking a break, which could be lethal to the cell, and fixes it, but it has the complication that it may not have done it in an accurate way. Okay? Sort of like, when I do a home repair. Right? I have a limited set of tools and a limited set of knowledge, and so I can repair it, but it may not have been done in the best possible way. Okay. All right, so non-homologous end joining, it's a very important pathway, and it really contributes quite a bit to maintaining DNA after it has been broken. But you have the potential there for loss of information. Okay, so then there's another pathway called single-strand annealing, and this is where we're going to think about complementary base pairing a little bit. So here we have our double-stranded DNA, and it's broken. And then what happens is proteins are going to chew back or recess those ends. And so you have the two strands here, and so one of these strands will be chewed back. Okay? And then on the other side of the break, another strand will be chewed back. And these yellow boxes represent complementary base pairing. And so what happens is you can chew it back to the point where you expose the single-stranded regions on the left side of the break and on the right side of the break where those can now base pair. Okay, so those will be complementary base pairs. And then once those pair, then these little flaps get removed, and then you have a gap, right? This is a single-stranded region here, a single-stranded region here. We want DNA to be double-stranded. It's most stable as a double-stranded molecule. And so then these gaps will be filled in, and then you have you've repaired the break. But what you can see is here you have two of those sequences. Now you have one, and so you have a loss of some information, right? Whatever was in between here is now gone. But the break has been repaired. Okay. So that's single-strand annealing. The process that we're going to talk most about today is called homologous recombination. And so homologous recombination is going to use another DNA molecule which has the same or very close sequence information and use that information to copy it and restore what is lost in the molecule that's broken. Okay? And so this is a more complicated process, but in general, this is what happens. Here we have our double-stranded DNA, and the DNA has been broken. And so what happens is enzymes come in. So these are proteins that will then chew back one of the strands. the strands will be coated by another protein that helps stabilize that single-stranded region. Single-stranded regions can be very sensitive to damage, or they can be chewed back by other proteins, and we don't necessarily want that. And so this protein will come in and preserve it, or help preserve it. And then what needs to happen next is a protein we're going to talk about a fair bit today, a protein called REC-A. So the REC stands for recombination. Okay, recombination protein A, because it was one of the first proteins found. And this will coat on that strand. And then here is the red DNA is our homolog. Okay, it has the same or very similar information. In the case of the bacterial cell, it's going to be exactly the same. And this strand will invade that other double helix. And then you have DNA synthesis, so now it's going to begin to copy that information. Okay? Likewise, the other strand will base pair with the second strand on the homolog, and then a DNA polymerase will copy that information. Okay? And this will continue. I know this looks a little complicated here, but you have these two DNA molecules that have now crossed over, and we're going through a process of copying the information from one to restore the information on the other. And then we get to this intermediate here, and then the DNA will be cut. And it can be cut in one of two ways. The bottom line here is that you have the DNA that was broken, that information has been restored because it was copied from the other one. And in some cases, if it's cut the other way, then you have an exchange of information between the two chromosomes. But regardless, the information that was lost due to the break is now restored in the, because it was copied from the other DNA molecule. Does that make sense? And so you're using the complementary sequence to copy it and then restore the information that was lost when the DNA was broken. Okay? And this is a process that is highly accurate. Okay, so one of the steps that we have worked on is really trying to understand this initial step. And this initial step has been worked on by a number of people, including my lab, in trying to understand how REC-A protein ultimately gets to this part of the cell. If you remember, I showed you that image with all that DNA. And if there's a break in there, when it's all packaged within the cell, how does REC-A get to that break site? And that's something that we have worked on quite a bit. And that's one of the stories I'm going to tell you this morning. So, what I'm going to show you is I'm going to provide some evidence that there's a complex called REC-FOR, and these are 3 proteins that will help REC-A ultimately form a complex on this single-stranded region after the break and allow for recombination to occur. Okay. So, the organism that we study is called Bacillus subtilis. And this is a picture of I guess the backyard of one of my graduate students. And it is a soil, it's found in the soil all over the world. Okay? And so it's also on the bottom of your shoes. And the reality is a lot of foot odor comes from Bacillus subtilis. Okay? But we use it because it's an incredible experimental system. Okay? And so we have many different experiments that we can choose from. And so we can ask a biological question, and we have lots of experimental angles to attack that question. It also grows very quickly, and so it will double in about 20 minutes. Right, which allows for us to do experiments very rapidly. It's less complex than multicellular organisms, okay, but it has a lot of the same pieces or very similar pieces to do the same kind of job. Okay? And so we can learn a lot about biology by studying a simplified system. And what we try to do, and what we usually are able to do, is to use Bacillus to answer questions that are fundamental to DNA replication and repair across biology. Okay, so we're going to take a particular type of approach here, and so I want to give you a sense for that approach. We're going to use whole cells. We're going to do analysis in whole cells. And so this is a relative size of organisms. And so this little dot here is the relative size of a virus. This is the relative size of a bacterial, sort of your average bacterial cell like E. coli or Bacillus, the organism that we study. And then this is the relative size of a yeast cell, like a baker's yeast cell, for example. And its DNA will be contained within the nucleus. And you can see that, I mean, look at the size of the bacterial cell relative to this. And so having a bacterial cell being small presents certain kinds of challenges, but we have ways of imaging certain molecules within the small cell. And so I showed you this, okay? And so what we can do is we can use fluorescence microscopy to label specific macromolecules within the cell and then see where those go or what happens, all right? And so I've already shown you that we can label the cell membrane with a fluorescent dye. We can label the DNA with a fluorescent dye. And we can also label specific proteins with another protein that is fluorescent so that we can see it in living cells. Okay? So there's a protein that was isolated from jellyfish many years ago, and within biology there's quite a bit of bioluminescence where where different organisms will need bioluminescence to do something, right? And so jellyfish often use a protein called green fluorescent protein to luminesce, all right? Fireflies use a different method to have bioluminescence. And so what science has done is take advantage of what exists so that we can study the proteins that we're interested in. And so what we want to do is take the green fluorescent protein and then fuse it to the protein that we want to study. Okay? And then we can see where it is in the cell. So the way that you do this in general terms is, so if we have a gene that we're interested in, so this is representing the DNA, then we go through, and it's very easy. This only takes a couple of days. Okay? I think my students would say it only takes a couple of days. Maybe it takes a little bit longer. But what they can do is take the gene for green fluorescent protein and fuse it to the gene of interest. All right? And so then when this gene is transcribed into its messenger RNA, then you have the message for gene A and the message for GFP as one. And then when that message is translated into protein, then you have protein A, which is now fused to green fluorescent protein. So they are one protein. Okay? And so wherever protein A goes, well, we'll have a chance to see it because it is now fused. All of those proteins from that gene are now fused to that green fluorescent protein so we can image it in live cells. Okay, so this is an example of that. And so here we're using Rec-A GFP, and so Rec-A is important for recombination. I have this is a black and white version, just in case you couldn't see it with green. But what you can see, and this is just a GFP image, then I have GFP overlaid with the membrane stain, which is in gray. And then next to that, for the same group of cells, I have the DNA and the membrane stain overlaid. So you can get a sense for where the protein is relative to the DNA. And so what you should be able to see is that in most of these cells, the GFP is fairly diffuse and diffusely distributed throughout the cell. It's not showing any particular location, all right? Now there's something here. It's like, well, okay, so I can see that forming, and so something's going on. There's a little bit of a complex forming there. Okay? Now what I'm going to do is take cells that are expressing this GFP fusion, and I'm going to treat them with something that will damage the DNA. Okay? So you should be able to see that there's a considerable difference now, where the protein was diffusely distributed in the cell, and now it's forming these distinct complexes, one in each cell. Okay? And if we look at where these complexes form, obviously the DNA takes up a large volume in the cell, but these do form, or they're coincident with the location of the DNA. Okay? So what we want to do is we want to use this as an assay or a method to understand how these complexes are forming, because this is triggered upon doing something to damage the DNA. We see this rapid relocalization of the protein, and we want to understand what is behind that, or what proteins are contributing to that rapid relocalization. And so the model that I'm going to present that we're going to test is this. And I've already discussed this a little bit, but basically we have a break, and I'm just showing you one side of the break, and that will initially be bound by this single-strand binding protein. And then the REC-O-RF complex will be important for REC-A to form a complex on that or at that break. Okay. All right. So we're going to use this assay, this fluorescence-based assay, this protein localization assay, to understand the underlying genetics behind that response by the cells. All right. So there's a fair bit of data here, but it's okay, we can walk through it. So what these cells are, I'm going to call the normal cells. And so they just have REC-A fused to GFP. We've done nothing else to manipulate the genetics of that cell. And so we can think of that as normal or our wild type control. Okay? And so if we don't, in this here, we're adding different chemicals or using UV light to damage the DNA. Okay? And so I don't want you to get too wrapped up into what these specifically do. I just want you to look at the response that occurs. And so if we don't add anything, then we see, and then we're scoring this as a percentage of cells with REC-A GFP foci. So what percentage of cells have foci or have localizations in them? All right. And so when we don't do anything, then we get about 10% of cells have those complexes that form. And it's because they're undergoing, we talked about normal cellular metabolism, and you can have reactive oxygen species and other things that will damage the DNA. So you're always going to have some level at which, growing normally, that protein is going to be responding to either a break or perhaps a problem with the replication fork. Okay? And then if we add these different chemicals here that either damage the DNA or block replication, and they do damage the DNA in different ways, what we begin to see is we begin to increase the percent of cells that have those complexes. Okay? And so, and again, it just, the increase depends upon how the type of lesion, the type of damage that is caused. Okay? So if it's there's some of the chemicals won't elicit the same type of response from the protein because of the way in which they modify the DNA. Okay. So if we think about that REC-FOR complex that I was talking about, well, what happens if we take the cells and we delete the REC-O gene? Right? There's really no response, even when we're damaging those cells, right? If we delete the REC-R gene, I mean, there's a little bit, but not much. We've really gotten rid of the ability of that protein to form those complexes. If we get rid of the REC-F gene, well, we do see a response. However, it's low, and then if we add DNA damage, it's not an inducible response. Okay. So at least in this particular experiment. And so what we learned from this initially is that REC-O and REC-R are required for that protein to form those repair complexes. Okay? So the breaks and the damage are there, but it's not able to organize into the repair complex without these two proteins being there. Okay? With REC-F, we're not sure, and so this is a snapshot, and so what we wanted to do was look at this in a time course. And it's sort of, instead of looking at one particular time point, we want to now look over time and see what happens. Okay, and see it's a better way to see if there's a temporal or a timing issue. Okay. And so this is what we did. So here we have time, and we're looking again at the percentage of cells with REC-A GFP foci, and this is our sort of our normal or wild type control with just the REC-A with the green fluorescent protein fusion. And this is treated with chemical 1, and it doesn't really matter what these chemicals are at this point. If we treat with chemical 2, then we end up, although there's a delay, it takes it longer to respond, we have roughly the same percentage of cells over time that will be responding to that type of damage. If we knock out the REC-F, so there's no REC-F protein, and then treat with chemical 1 or chemical 2, then it will increase with time, but it never reaches the point of what we see when RecF is there. And so it's an efficiency issue, right? It's sort of like if I'm driving my car somewhere, if my wife is with me, we get there in an efficient amount of time. Okay? If she's not there, I still get there, but it may not have been efficient, all right? And so RecF is providing some efficiency and allowing for Rec-A to form those complexes in response to damage. Now, one of the issues that we always deal with is you treat with a chemical and you're treating living cells. And so you don't really know what's going on inside of that cell. You don't really know, are we getting breaks? If we're getting breaks, how many breaks are we getting? What other kinds of damage are being created? We don't have it's very difficult to quantify what is occurring at the DNA level. All right? And so to go further with this, we chose a different approach. And so what we want to do is just make a double-stranded break in the DNA, okay, and that's it. And so what we can do is we can take our bacterial chromosome here and we can put into the DNA a recognition site, so a DNA sequence that will be recognized by an enzyme, and that enzyme's job is to cut DNA. Okay? Then what we do is we take the enzyme and we put it under control of, we can control its expression. And so we can add a chemical that will cause that enzyme to be expressed. And once it gets expressed and made, the cell makes it, then it will find that DNA sequence and cut it. Okay? So now we have a controlled way to make one double-stranded break in the chromosome by expressing this enzyme and having that recognition sequence in the chromosome. And so now we can go back and test these different genes, and to determine what their effect is on specifically repair of a double-stranded break. Okay? So we did that, and here is our wild-type control. So we can see that as we induce the break here at time 0, and within, you get about I'm on an angle here, but roughly 70% of cells are showing a response within whatever that is, 80, 90 minutes. Okay. If we knock out RecF, you can see that it's still responding, but it's very delayed in that response. If we remove Rec-O and Rec-R, there is no response. Okay. So we've found that regardless if we're using a chemical or if we're making a site-specific cut where we have that enzyme that would just come through and cut both strands, you need, for sure, Rec-O and Rec-R to get a response, and Rec-F is very helpful to the efficiency of that response. All right, so we're going to come back to our model here, and we're going to do one more test. But what we were able to do in thinking about the data that we found, as well as data that other people had published, that once this break is coated by single-stranded binding protein, then Rec-A ultimately needs to displace it. Something that is interesting is that this single-stranded binding protein, if you notice I have these little tails here, and those will interact with other proteins. And so it will interact with this Rec-O protein and then help bring that complex, bring those proteins to that site. Once they're there, then Rec-A will replace SSB, and then it will coat that end of the break, and then it can go through that homologous recombination event. All right. So what I'm going to finish up this little portion here, what I'm going to tell you, so one of the nice things about the genetics that we did is Rec-O, Rec-F, and Rec-R, those genes are not essential for viability to the cell. So you can knock them out and then study the effect. Okay? SSB, single-stranded binding protein, is an essential replication protein. And so if we were to try to knock out that gene, the cell would die and we would be unable to study it. Okay? And so what we were able to do was use a variant of that protein that allowed for survival but was impaired in doing this step. And we were able to then show that SSB is important for the recruitment of these proteins. Okay. All right, so now what I'm going to do is we have a bit of background now in DNA repair. And we're thinking about double-strand breaks and so on. And what I want to do is provide you with a couple of examples in nature that are very interesting, that I find very interesting, and I hope that you'll find them interesting as well. So there's an organism called Deinococcus radiodurans. Has anyone heard of this organism? No? Okay. So Deinococcus radiodurans, I mean, it's pretty common. It's like bacillus. It's in the soil, all around the world. And this is just an image of it that I borrowed from the internet. And it's very, very resistant to ionizing radiation. Okay? Very resistant to ionizing radiation. So let me tell you just a little bit about ionizing radiation. So ionizing radiation, in general terms, it has enough energy to ionize a molecule. Okay? And so basically what that means is it has energy that can remove an electron from an atom. And there are two basic types of this. And there's electromagnetic, which comes in the form of gamma rays and X-rays. And then there's particulate, which comes in the form of alpha and beta particles. And so what happens with ionizing radiation is, this is sort of a simple representation, but you have a gamma photon, and if it interacts with an atom, then it can cause an electron to be removed. Okay? There's enough energy to do that. When that electron is moved, if we're inside of a cell, we are in an aqueous environment. We're in a water environment. Okay? And electrons react readily with water, and they will form hydroxyl radicals. Okay? And then it is this piece here, it's this hydroxyl radical that can damage cellular material. All right? So it can damage DNA, RNA, proteins, lipids, carbohydrates. Okay? And we're going to focus on the damage of DNA and proteins. All right. So when we get into this, I want to sort of orient you a little bit to radiation doses. And so these are approximate. I don't have exact numbers for this. And so, just keep in mind these are approximate numbers. But I think the average chest X-ray or so on, you would be exposed to less than 1 milligray, okay, as a unit. A person who would be exposed to 5 grays, that would be a lethal exposure, a lethal dose. Okay? And then, just for comparison, I think for many of you, you're well aware of Chernobyl, which was where there was a reactor meltdown and there was radiation released. And people who were, and I don't have great numbers, so I can't find great numbers for this, but people who were in the immediate vicinity of that probably had exposures on the order of 0.2 to a little less than 20 grays. And so that's, to get exposures at that level, you're dealing with a sort of a reactor meltdown type of situation. All right. So now let's take a look at Deinococcus radiodurans. So these are kilograys. So this is 2,000 grays. This is 4,000, 6,000, 8,000. E. coli, and this is surviving fraction. E. coli, a culture of E. coli is essentially sterilized once you get to about 1,000 grays. It's certainly sterilized at 2,000. Okay? Deinococcus radiodurans at that dosage will actually be growing and doubling. And then until you get to around the 4,000 gray mark, 6,000 and so on, that's when you begin to see quite a bit of cell death. Okay? So it can withstand a tremendous amount of radiation. Okay. And so for comparison, if we were to take what is called the the D37, or the way to think about this is the fraction, or the dosage at which 37% of the cells will survive. For E. coli, with growing exponentially, will have about 4 to 5 copies of its genome per cell. You would get about 8 to 9 breaks in a chromosome. Okay, and then only at that level of chromosomal breakage due to the ionizing radiation, you would have, only 37% of those cells would survive. For Deinococcus, its 37% survival is, you're getting about 275 breaks per chromosome. Okay? So again, it's able to repair these breaks very well. All right. So then the question that people wrestled with for a long time, and are still studying it today, how does it do it? Right? How can it incur that amount of DNA damage and still survive? Right? So, I'm sorry, this is a copy of a copy, so it's not very crisp. But it has the same types of DNA repair machinery that you find to be among most bacteria. And so, for example, this is just another measurement of radiation, but here we have Deinococcus, and then eventually, this is a killing curve where you get cell death. And then if they knock out the REC-A gene, which we just talked about, then you can see that it's highly sensitized, right? Because it's using REC-A for most of its repair. Okay. Now, interestingly, though, if you knock out the REC-A gene in that organism, it still survives better than E. coli when it has it. Okay. So it has, like many types of cells, they have other pathways that can be used. And so what happened, people began to think about basically the repair mechanisms that occur, and they began to propose a model. And so what happens is the chromosome, and there's many contributing factors to this, but ultimately what occurs is you can have the chromosome that's broken into a number of pieces. And then we talked about proteins being able to chew back or recess that DNA, right? And so that's what you're seeing here. So each one of those pieces can then be chewed a little bit, which will expose a single-strand region. Then what occurs is that single-strand region, just as we've seen in homologous recombination, can be used to invade an intact chromosome. And it can then, you would go through a time of DNA synthesis, and those pieces then get extended. Once those pieces have sufficient homology, meaning that there's complementary base pairing, right? So you basically extend them, and then once you have complementary sequences between the different pieces, then those can then base pair. So we go through. It can then, this now looks a little bit more like single-strand annealing here, where all of a sudden these pieces can then anneal together, okay? And then it can go through and then remove the flaps and then fill in the gaps and restore the chromosome. All right. And so is this type of repair really any different from anything that occurs elsewhere in biology? Does it have different proteins that do it? And the answer, for the most part, is no. It does not. All right. Something else that I need to mention is that Deinococcus is able to, so when you have ionizing radiation, you have all these reactive oxygen species that are generated. There are a couple things that happen. One is the DNA is damaged, right? The other is the proteins that are responsible for repairing the DNA are also damaged. And so Deinococcus is able to limit that protein damage so that it can go through and do those repair pathways. And so it has a number of different strategies to limit the amount of protein that is oxidatively damaged due to those reactive oxygen species. All right. So we've gone through and we've talked a little bit about doses, and so you know that a bacterium that's in the soil and is on this planet is not going to have to deal with 2,000 grays of radiation. Okay? So what is the advantage? Why does Deinococcus have the ability to withstand and to basically withstand this type of damage and survive it? What is the evolutionary advantage of that? I want you to think about that for a moment. And so the consensus for that is this, desiccation, okay? So what happens is, this is out in the environment, right? And we all live in Michigan, you've seen the weather, right? It can be very dry and it can be very wet, right? And Deinococcus is very good at surviving a lot of stresses. And one stress that has a lot of similar features to being bombarded by ionizing radiation is actually dehydration, okay? And so it turns out that Deinococcus is very resistant to, the way we term that is desiccation. Okay, you can really dry it out and it's very resistant to that, and desiccation results in hundreds of DNA breaks within the chromosome, okay? And so the reality is with a lot of things that we test in the lab, or other people test in the lab, we use something and it gets repaired, but there's probably some other more relevant way in which that damage will occur, right? And in this case, for Deinococcus, it's probably desiccation. It's also resistant to a lot of other stresses as well. Okay, so in the last 7 minutes or so, I wanted to choose another aspect of biology that I think is very interesting. So hopefully you enjoyed the little Deinococcus story here. So I'm going to try one more little story, and then you can go off and watch a football game. All right. So this is a fungus or fungi, Cladosporium, and it's just here in this image. It's grown on 2 different types of medium. And all I want you to take from this is that it has a black coloration to it. Okay? And that black coloration is melanin. Okay, and this is something I just learned about. People have been studying this for a little while, but I learned about this summer, and I thought this was really cool. And so melanized fungi, or fungi that are able to make melanin, inhabit some very extreme environments, including the Arctic, Antarctic, and very high altitudes. Okay, so I mentioned Chernobyl, so I'm going to mention that again. So the Chernobyl accident occurred in 1986, and then in the early 90s, several groups, including this one, sort of investigated what was growing in the soil around Chernobyl. And what they found was a very large fraction of the fungi are melanized. Okay? And they even found melanized fungi growing on hotspots of reactor 4 that melted down. Okay? It's pretty sweet, right? Gotta love biology. So then people have also done studies on both the International Space Station and Mir, where they're looking at microbial contamination, and they find melanized fungi there as well. Okay. So then that sort of begs the question, how does melanin contribute to these fungi being able to inhabit extreme environments? So I want you to think about that we don't have a lot of time. Let's just think about that for 30 seconds. And so you may be able to come up with a couple of hypotheses to explain that. Or none, that's okay too. It is early on Saturday morning. Okay, so the hypotheses that people came up with initially are that melanin, which is a pigment, may provide some type of shield for the cell. Okay? And that might be able to shield the radiation or something along those lines. And for some fungi, that's probably the answer. Okay? And then the other possibility is that perhaps melanin could harness the radiation as a source for energy. And so it turns out that this became tested very rigorously. And I'm going to show you a couple of those experiments that people did. So here we have a radiation source, and then here we have a melanized fungi that is growing, and they're germinating. So these are spores which are then transitioning into what's called hyphal growth, and that's these protrusions that you see. And so what these researchers did is they measured the angle for which those hyphae grew relative to the radiation source. And so the bottom line is, if you have a small return angle, then it's growing towards the source. If you had a larger return angle, right, if it's growing in this direction, a larger angle, it's growing away from the source. And and they did this for a number of melanized fungi, and they found for many of them a very short return angle, indicating they were growing towards the source of radiation. They then did a number of other experiments, and I'm just going to show you one, and a number of different controls. And I'm not going to show you any of those. Because we only have 3 minutes now. So, but this is a measure of growth. And so we have the melanized fungi that is not irradiated and the melanized fungi that is irradiated. And you can see that there is an increase in what is colony-forming units, or an increase in growth. All right. So the idea here is that it's able to take gamma rays and then take that energy and then have the cell convert that into chemical energy, right? And so are there other examples in nature where electromagnetic radiation is converted into chemical energy? Right? So, photosynthesis. Plants have pigments called chlorophyll which gather visible light, and that allows for electron transfer to electron-carrying molecules. And if we think about the electromagnetic spectrum, right, it goes from gamma rays to radio waves. Chlorophyll harnesses visible light, and these fungi appear to be harnessing gamma rays. And they've been able to show that there is indeed it is stimulating electron transfer both in a test tube as well as in the cell, and that you are getting an increase in energy-carrying molecules when those cells are irradiated, and it depends upon melanin. Okay. So that is another example. Then, in my opinion, that's really cool, right? I mean, how biology is able to. I mean, it's different, but it's a variation on a theme. And it's able to then use melanin to harness gamma rays to ultimately convert it to chemical energy. So the bottom line here is, in biology on our planet, there's a lot of very interesting examples of survival, and there's very interesting ways in which DNA can be repaired. A lot of that is conserved. Right? And there are ways to harness energy, and although there are variations on a theme, a lot of the themes are very similar. So with that, I would like to thank you. I would also like to thank my research group. And so the research of mine that I showed was done by Justin Lenhart, who is here. We are funded by the National Science Foundation and the National Institutes of Health. And we have our research is carried out by a postdoc, graduate students, and undergraduate researchers here at Michigan. So thank you.